The overall goal of this experiment is to observe the effects of compressive loading in the mandibular condylar cartilage of mice, using morphometric measurements in radiographs and histological analysis.
These methods can help answer key questions regarding the structural and cellular changes within the mandibular condyle of rodents.
The main advantage of the techniques shown here is that they could be used to analyze other small bones of experimental animals.
For this protocol, have isolated, dissected mouse mandibles ready for use. On a petri dish, transfer the mandibles into an X-ray cabinet system. Then, take radiographs at 26 kilovolts for 5 seconds. Now, open the images in the analysis software to make morphometric measurements. First, use the unit button to set up the scale bar. Next, select 6 anatomical points using marker style 2. First, select the most posterior point of the mandibular condyle as point 1. Next, select the incisor process for point 2. Then, select the deepest point at the sigmoid notch for point 3. Next, select the deepest point in the concavity of the mandibular ramus, point 4, and then, select the most anterior point of the condylar articular surface, point 5.